Okay, yeah. Okay, so it should be recording now. Yeah. Okay, so yeah, so welcome, as I said. And so these KCN events were initiated back in 2012, as many people of you guys know. And really the goal here is sort of to bring together researchers, trainees, Kreml, and the wider community to really discuss, aspects in the fields of computational mathematical neuroscience. And really what we want to do is sort of raise awareness in recent advances in theoretical computational neuroscience, help catalyze new collaborations, just make people aware, reveal new research directions, local groups, emerging established leaders in the field, and really give people the opportunity to share the vision in sort of a sort of relaxed, informal sharing fashion where there's lots of discussion. Of course, virtual limits that to a certain extent, but we'll do the best we can. And really to sort of, bring things together because we need all these sort of, efforts to be able to, tackle the challenges of the brain. So computational neuroscience, as you guys all know, is sort of a very wide and diverse field. And there's obvious clear overlap with neuroinformatics, which is becoming more and more important these days, more important and critical with these big data. And today, I'm very pleased to welcome Dr. Etay Hay, who's one of the independent scientists at the Kreml Center for Neuroinformatics at CAMH. He's an assistant professor in psychiatry and physiology, and he obtained his PhD in neuroscience at the Hebrew University of Jerusalem, and then went on to do postdocs at Baycrest and University of Western Ontario before starting up his own independent lab. And so, as some of you may have read, he's interested in using, well, he does use computational models of cortical microcircuits to study mechanisms of brain processing in health and mental disorders. And recently they've been developed these human cortical microcircuit models, which I'm looking forward to hear about. And so without further ado, Ita, if you want to put up your first slide, you can go ahead. What I would say is just to let people know, so this has been recorded and it'll be online after the KCN Hub site with Itai's permission. If you have questions during the talk, Itai is happy to answer them. But what I would just say is just put it in the chat or put your hand up because I'll be monitoring it. And, of course, any, immediate questions, feel free to ask. Itai is happy to answer them. But if you have more detailed, extensive questions, please keep them to the end because there'll be lots of time for discussion. And if people want to sort of keep talking and have, additional discussion points, Itai is more than happy to stay around. I'm very happy to say, but I'll sort of formally end it at some point for people to go. So yeah, so that's the sort of technical aspects. So without further ado, welcome again, Itai, and the floor is yours.
Thank you. So you can see my slide, okay?
Yep.
All right, so thank you for having me. It's a pleasure. I think this journal club is really cool. And yeah, so today I'll talk to you about the work that we've been doing over the past year and a half or so in my lab to study the effects of reduced inhibition in depression on cortical processing in human neuronal microcircuit models. So we are studying the cerebral cortex at the scale of microcircuits. And that scale spans between a whole column usually, which is like about 30,000 neurons in the half a millimeter by half a millimeter by a millimeter of the cortex. To also just sub-networks in layers, for example, in layer 2-3, you have a few thousands of neurons. And those neurons have distinct types. There are a few key ones, and recently the Allen brain is even talking about hundreds of types, subtypes. And also those neurons have distinct connectivity that we'll see plays a role in their functionality. So a bit about the kind of processing that is happening in those microcircuits. Obviously, there are things that take many regions of the brain together to do, but small networks of thousands of neurons can do quite a bit. And for example, they extract features, and these can be as simple as edges, visual edges of a certain image, or a sound in the auditory cortex. And it can also be more complex features like parts of an object or even a whole object. Depending on where you are in the cortex, the function or the extraction and the processing that is done in those circuits differs in complexity. But conceptually, there are quite a lot of similarities in looking at all those processing functions. So what's interesting, you may know from artificial neural networks and deep learning that has gained quite a um, fad in the recent years, and for a good reason. I think there is interesting things that the artificial neural networks field can show us. When you have a bunch of neurons and you learn their specific connectivity, you can do a lot of interesting things. So for example, you have like smart machines that can recognize images or digits, help you translate. And similarly in the cortex has, I would say, two powerful things going for it. The first thing is the connectivity. So you have very fine-tuned connectivity that can enable a network or a microcircuit learn how to do something. Also, the physiological neurons are a little more complex than their simple artificial counterparts. There are similarities, but also it's best to view those as electrical entities that have three-dimensional structure and properties in different parts of the neuron, the soma, the dendrites, and those nonlinear properties and distributed properties give them a bit more power in computing things. So when we study microcircuits, we're looking at the function and how those mechanisms contribute to those functions. And specifically in our lab, we're looking at the focus of these things in mental health. So the power of looking at those biophysical and detailed models is that they allow us to test certain hypotheses about changes in disorders that involve mechanisms, either in cellular or circuit mechanisms, and try to characterize their effect on cortical processing. And currently, this is something, especially in humans, that is quite hard to do experimentally. Most of the microcircuit to function kind of characterization work is done in rodents, sometimes in monkeys. So that's another thing where computational models are a very useful tool for those questions. So in my lab, we pretty much focus on depression. We have new project looking at schizophrenia as well, using similar approaches, and also some projects looking at aging. And interestingly, there is some overlap between depression and aging in some of the mechanisms and the effects. So that makes for an interesting um, bunch of projects to do and then compare. So we look at there are three central themes. One is to study the mechanisms of cortical processing in health and mental disorders. Another thing is to simulate clinically relevant signals such as EEG to try to identify biomarkers in the EEG that correspond to those effects that stem from the microcircuit changes. And lastly, we also use our computational models to test new pharmacology in silico. And I'll mention the last two more briefly. I'll focus more on the first theme of studying processing in health and depression. All right, so the beginning of the project, it really stemmed from a few interesting findings, more recent findings about changes in depression in those building blocks of the microcircuit. One of the cell types called somatostatin interneurons, or SST, inhibits the distal dendrites of pyramidal neurons, as you see here in the schematic, and very selectively. So what people, first of all, they play an important role in computation because of that. And I'm not going to elaborate too much on this here, but what's interesting is that our collaborators, the Sibyl lab, Etienne Sibyl at KMH, found in post-mortem tissue from healthy and depression patients that there is a reduction in SST expression in SST interneurons. So they were able to look at the per neuron, essentially, which is a very nice resolution of examining gene expression. And across all the cortical layers, they saw about a 40% reduction in the SST expression, and that's an indicator that there is some malfunction in those cells, presumably a weaker inhibition. So also as a note, I'm referring to major depressive disorder here as depression for simplicity for the rest of the talk, but keep in mind that we're studying the severe type of depression, which is a leading cause of disability in Canada and worldwide. But yes, in this presentation, I'll just refer to it as depression. So another interesting finding that supports the role of SST reduction in inhibition in depression is that there are drugs that boost that inhibition and then lead to antidepressant effects. So the same lab, the CB lab, worked on engineering drugs that are more specific in boosting the receptors, the alpha-5 GABA A receptors that SST interneurons target. As you can see here, the effect of the drug in red compared to the no drug in black is larger for alpha-5 receptors than alpha-1. Alpha-1 are distributed across both basal and apical dendrites of the pyramidal neurons and also other interneurons. The alpha-5 are more in the apical dendrites. So when they, that's the more cellular characterization, and when they injected those new drugs, which are called alpha PAM. PAM stands for positive allosteric modulators of the GABA A receptors. So whenever there is GABA around, they will enhance the inhibition mediated by those receptors. And when they injected it to mice that underwent a stress protocol that is established model for depression in rodents, then when you measure how well, if they're exploring the maze more like control, so this is control level, alteration rate is sort of like when there is a Y maze. I don't know if some of you have heard of, there are branches of that maze as unhealthy, or I would say non-depressed individuals would explore more and turn to alternate more between those arms of the maze. The depressed one would not, which is here. And then as you give more and more of the drug, you recover that ability. And so it is a very promising drug for treating depression. And also, at least mentioning it here, it is a further support of the role of SST inhibition being reduced in depression. And I'll mention a bit more about the drug at the end in relation to some ongoing projects that we have with Etienne Sibyl and Taufik Valiante from Krembo. So those SST interneurons, as I said, mostly have been characterized in rodents because that's the kind of animal that we have a bit more access to the circuit components. And interestingly, they tend to be active more during baseline before stimulus. Here, the dashed line is the time of the stimulus. Those are behaving rodent recordings awake in vivo. And as you see, the rate of those SST interneurons is higher at baseline and less during the early phases of the response. And this is like a brief whisker stimulus, and you see a reduction in the rate of those cells. So the idea is that they tend to play a bigger role in maintaining baseline activity. You can see also that, the different types, it's worth mentioning them now. Obviously the pyramidal in the bottom here is the key excitatory cell type. And then you have three key interneuron types, the SST, the PV, that tend to be a fast spiking neurons, and the VIP that tend to inhibit neurons and provide disinhibition in the circuit. So you can see each type has a typical rate pre and post stimulus. But for our story right now, the SST finding is very interesting. And that leads to the leading hypothesis that a reduction in inhibition, and we're talking about both the synaptic inhibition that SST provides and also the tonic inhibition that is mediated by other receptors, but also is like the sum or the effect of the variety of interneuron inhibition, and SST contributes to it. So reduction in those types of inhibition would lead to a more noisy baseline activity, because like when you have the intrinsic spiking in health, it tends to be lower in pyramidals. And if you reduce the inhibition from SST, you will get them to fire more. So they become a bit more noisy compared to signal. The signal should not change so much because SST don't play so much of a role, at least in the early parts of the response, the cortical response. So that leads to the idea of a reduced signal to noise ratio and a reduced detection quality. And yes, so before I'll tie to some other aspects of depression, I'll just mention that this is relevant to both detecting a stimulus, yes or no, or also extracting feature. This is really an expected deterioration of the ability of the cortical microcircuit to do its job, essentially. Yeah. And in depression, this increased baseline activity is thought to underlie the symptoms of rumination, where patients are more thinking inside their head and more unable to stop their inner thought loops. And also there are studies that show a reduction in signal detection quality and attention that go along with that. So, having that as a background, we wanted to test this hypothesis and characterize things a bit more mechanistically, because as I said, it's really hard to do it in vivo, in humans especially. So, in recent years, there has been increasing data of human cellular and circuit electrophysiology and anatomy. And so it really opens an opportunity to constrain data-driven models that can help us to tackle some of those questions and characterize effects. So the first stage was to model single neurons of the four key types. And there were available models, but they didn't capture some of the properties as well. And so we, because we have experience in fitting single neurons, we just fitted new models ourselves. The pyramidal data is taken from our collaborators, the Taufik Valiante lab. And the interneuron data is from the Allen Brain database, which is a public open database, very commendable. You can just go there and download things. There is a lot of annotation about the data. So the morphologies we use from the Allen, but the pyramidal electrophysiology data are from the Taufik lab. So here you see the model performance for each cell type. Let's start with the pyramidal. So the models, those are L2-3. We focused on layer 2-3 because that's where most of the human data comes from. And that's pretty much a well-defined entity in terms of processing. Obviously, there is a lot going on between layers as well. And our plans are definitely to expand this. But for most of this talk, I'm going to talk about L2-3 models. So this is a human L2-3 pyramidal. Here you see the firing in the model and the FI curve. So the relationship between how much input you inject and what's the rate that the neuron fires at. So in gray, you see the experimental range and also the mean. You see that the model is quite well matching that profile of the statistics of the experimental population. That's something that the Allen brain models didn't do too well for a variety of reasons that are probably beyond the scope of this talk, but I'm happy to get into if anyone is interested. But yeah, so we reproduce the FI curve, which is a very important property of those cells. It's really the gain, the gain of their firing given an input. In addition, we also capture an important property that is called the sag voltage. That's when you give a hyperpolarizing step current, that little blip you see in the beginning is a result of a dendritic H current that is mostly in the apical dendrites. So again, the Allen models did not include any channels in the dendrites, and we felt we definitely need to constrain the dendrites a bit better. So we both constrain some of the passive properties of those cells, like their membrane time constant and other things, but also the sag voltage, which differs in different cells and plays a role in a variety of properties. Actually, Scott and Homera and Taufik, and I think Francis might have been on that paper as well, just published a very interesting paper about it, which you may have heard of. So that's for the human pyramidal. Then for the SST, again, this is a reconstructed human morphology, and the electrophysiology comes from one putative SST interneuron human from the Allen brain. We again capture the FI curve, the sag voltage curve. And yes, that's, I think, all I have to say about this. And we also fitted PV interneurons, which are the phi spiking, as I mentioned before, the VIP that provide inhibition onto interneurons. And again, we match the experimental FI curves and voltage, sag voltage fairly well. So now that we had our single neurons, we set to fit some synaptic human properties. And it's, again, interestingly, there is, in the past decade, there have been quite a few papers characterizing human synapses. So that's really valuable data that is really available from the literature to fit. And in, I would say, the past couple of years, this synaptic data is being even further increased by high yield paired recordings in human tissue. I can also mention, in case some of you don't know, that that human tissue is mostly healthy tissue that is extracted during surgery, usually either in epilepsy patients or someone that has a tumor. So to get to the point of the epileptic focus or the tumor, you go through healthy cortical tissue. This is resected and then can be used for electrophysiology. So yeah, one of the key things, especially for our story, fortunately, there was a study that characterized the disynaptic loop that is mediated by SST interneurons in humans. And this loop has been characterized in rodents about maybe a decade before that paper. And interestingly, you see similar properties, and maybe that's a good time to maybe actually after I go through the components, I'll mention some of the similarities and differences between human and rodents.
Sorry,
Go on.
there's a question from
Yes.
Scott. If it's a quick one, Scott, go ahead.
Yeah, no, this is really quick. Etay, I'm just sort of curious, when you were fitting these individual neuron models, what was the justification behind focusing on just the IF curve and the SAG?
Right, good question. So first of all, I'm just showing those, but we capture a lot of other things. So I'll mention them and also say maybe some of the things we don't fit. So we those models reproduce the pattern of each cell. So for example, SST tend to adapt. You see the ISIs in the beginning are shorter than the later ones.
Yeah.
So many features of the spike, the pattern we reproduce, the time constant, and some input resistance. So all of those properties. Yeah, so I'm just showing this and also showing the SAG. Obviously, there are a few things that might be missing. I mean, you're welcome to ask about something specific that I may omit, but before that, I'll just say, obviously, there are some back-propagating action potential in the dendrites and calcium spikes. Some of you may know that I've fitted those in the past. For now, the data about it from human is not so conclusive and so good, and also, they make the simulations heavy and the whole fitting procedure is a bit more complex when you expand to those properties. So we didn't go down this route, but definitely that's something we're looking to do. Is there any property in particular that you wanted to ask about?
No, I was just like, I saw the adaptation and stuff. So I was
Yeah.
curious that you actually fit that. And so I assume you used a sort of similar like feature fitting mechanism that you did in your previous papers.
Yes, exactly. So we use the two types of algorithm just due to chronological reasons. I mean, Kent, which is on this paper, which I should mention has been submitted as an is on bioarchive, so definitely available out there for you to look further into. Kent fitted those two cell types using my old version of the algorithm. And then Alex fitted these two cell types using the BlueBrain package. BluePyOpt. So yeah, I mean, but it's quite similar for our purposes, but yeah, so we use multi-objective optimization algorithm. This is automated. It's very data-driven. You put in the mean and the standard deviation of the features, each feature like spike height, spike rate, and then you try to get, especially here with, fortunately, we had a lot of data from Taufik's lab. We have a population of neurons that because there is a diversity in the properties even within a cell type. So we use them to guide and judge the model, essentially, right? So yeah, that's exactly, yeah.
Awesome, thank you.
Yeah, so I'll mention here already, like at least in that scale, that the difference between human and rodent models, first of all, is their size. And I would say that's one of the key differences that leads to some, let's say L5 pyramidals or even L23 being double the size of the rodent counterparts. And that there have been a few studies that showed that the distal parts are more isolated electrically from the soma in humans than in rodents. I think there is still work and analysis to be done on that, and we might be looking to get into that a bit more, looking at the dendrites, but that's already one difference. So essentially inputs that part of the cell sees will be further away in humans than in rodents. So that's one key thing. In synapses, I already would say that they're really strong synapses, inhibitory, especially in humans. They're more reliable, there is less failure. So there is definitely a reason if you want to model human depression and human microcircuits to take that into account. So we use that values and data from the paper to fit what we call the disynaptic inhibition between pyramidals. So what happens is that, and this is the simulation that reproduces what you see experimentally, when one pyramidal fires a high frequency train, about 100 hertz, it excites SST interneuron that it's connected to. Here you see the connection points, the synapses in black. There are multiple ones. And because the EPSPs are facilitating, so essentially they tend to sum up much more than otherwise. There are three kinds of summations. One is the regular, sort of passive. One is depressing, where you sum, but you're even, whenever you sum EPSPs, you would tend to build and build and build. But when you depress, you do it less than the normal. When you facilitate, you tend to do it more. So the summation is higher. So in this connection, one of the very key things about it is that the summation is facilitating. And one connection being active is enough for this SST interneuron to fire. And that's not that common. I mean, some of you may know that usually it's necessary to have inputs from multiple cells, tens if not hundreds sometimes, to generate firing. But here it's a very powerful lateral inhibition mechanism in the cortex, where one pyramidal can trigger spikes in the SST interneuron and then inhibit neighboring pyramidals. And here again, you see the connections from the SST interneuron to the pyramidal, the neighboring pyramidal. They're usually on the apical dendrites, as we mentioned, and they generate inhibition. So we reproduce several things here. First of all, this whole setup, and in particular, the facilitating magnitude of the summation in SST, and also the amplitude of the inhibitory PSPs in the pyramidals. And yeah, so again, there is a high, relatively, I mean, you see the average is about 1 millivolt, minus 1 millivolt, which is pretty strong compared to rodents. There is low failure rate as well. As some of you may know, synapses are not that reliable always. In rodents, sometimes I think the failure is about, I don't know if it's the failure or success actually, but I think the failure is 30%. So like 30% of the time when you have a spike, you may not have a PSP in the synapse. But obviously because you have multiple contacts, then on the whole you see a response, but it's not always of the same magnitude. Here you see a few trials in our simulations that illustrate that. So we fitted this type of synapses, and similarly, yeah.
Etay, sorry, just quick interruption.
Yes.
Maliha has a quick question, I guess. Go ahead, Maliha.
Yes.
Hi, Dr. Hay. So my
Hi.
question is, so you mentioned just now that the connection between the pyramidal and the SST was very strong. And in your model, was that just a consequence of you trying to fit what you had from the experimental data with the curves in the red that you have, the spikes in the red? Was the strong connection just a consequence in your model of trying to fit to
Yes.
that?
So if I understand your question correctly, we had the finding in the paper and we reproduced them. So we constrained the model to reproduce. All these are properties that have been reported and we reproduce them, right? So the means to do that are, we don't need maybe to get into too detailed, but we do tune the conductance of each type of connection, for example, pyramidal to SST or SST to pyramidal. And so we tune that. We also tune the failure rate. We have models of synaptic synapses that are probabilistic and have all those parameters. So yeah, you would say that we constrain the models to reproduce the experimental properties.
Thank you.
All right, so we use similar approaches to fit a variety of other human synapses. Unfortunately, there is quite a bunch of data there. Pyramidal to pyramidal human synapses. There is a paper, also pyramidal to SST to pyramidal. That's the one I mentioned, and also pyramidal to PV, PV to pyramidal. So a lot of the key connectivity in the circuit, and here you see the 4 key types, is actually pretty well constrained by human studies. Some of the other connection types, we use the available parameters and values from rodents. So that's especially the VIP connections and some of the interneuron to interneuron connections. In general, both SST to pyramidal and PV to pyramidals in humans are stronger and more reliable, as I said. And recently, actually, there might be a bioarchive on that soon. There are new studies that show that also the pyramidal to pyramidal are stronger, but it still remains to be assessed. But there are indications that holds also for excitatory connections. So we reproduce the PSP amplitudes and the failure rates mainly, but also some of the short-term dynamics, which I refer to like the facilitating summation. All right, so now that we had some of those building blocks, the next step is to connect all those single neurons and their synaptic properties into microcircuits. So the first step is to look at the proportions of cell types in the human microcircuit. There are also nice studies about that, some post-mortem ones and some more recent Allen brain data, and also I think from Boston. I think in Harvard, there is a group that published a very nice paper that showed that the different proportions of interneurons in different cortical areas in across species, essentially. So they show that there is in somatosensory or more sensory areas, there is about 20% interneurons, so 80% pyramidals. In the prefrontal microcircuits, there is a higher proportion of interneurons. And they also show that there is a somewhat higher percentage of interneurons in humans compared to mice. It's not a huge effect, but it's there. So yeah, that's something that is interesting. And to remind you again, we're talking about L23. In our circuit, we had 1,000 neurons. Usually the amount would be more like between 5 and 7,000, but mainly because of computational reasons, runtime of those fairly detailed models, we have a representation of that including 1,000 neurons, but with the right proportions. You can see it here in this pie chart. So obviously most of the neurons are pyramidals, and the proportions of the different cell types are roughly similar, but there is definitely more VIP and PV in layer 2-3. Right, so the next step is, the next step is to do the connectivity. So once we have those 1,000 neurons, we need to connect them with physiological probabilities. And that's definitely not all-to-all connectivity, as we call it, like 100%. It's more around the 10%. In humans, it's a bit higher percentage. It's about 15%, according to the study I mentioned. Other types, other probabilities between the different cell types we took from mostly rodent data, because currently human data is unavailable, but they're also very type specific. Right, so after we connected the microcircuit, we give it a background input, random, that represents the kind of ongoing drive that it will from either thalamus or the cortex. And we tune two things to get the baseline firing right. One thing is that magnitude of this drive in the different interneurons and neurons, and also the connectivity a bit, because the connectivity is not so well constrained. We do have ranges, but we can tune them a bit if necessary to get the right firing rate and properties of baseline firing. Here you see a raster plot example of our resulting microcircuit. So before I get to some metrics, you can just see that those neurons tend to sometimes have more synchronous event. And here every color, every tick is a spike and color coded by cell type. Here are a few example traces from the different cell types. So you see obviously the PV tend to fire quite a high rate. The SST as well, as we know from the studies that the rate of those cells is high in baseline. Pyramidals tend to fire around 1 Hertz or a bit below, between like 0.5 and 1 Hertz.
Etay, quick question on that slide, the previous one. So the baseline firing, so just in terms of what you're matching, this is like from human single unit firing, like when you...
Yeah, that's the next slide will tell you.
Sorry,
yes,
go ahead.
No problem. Yes, so the few metrics that we constrain those models with to reproduce are the baseline firing rates. And for this, fortunately, there is some data in humans. So for pyramids, we have data extracellularly from humans. So usually this would be multi-electrode array or even single electrode array, even extracellular, just recording those spikes. So we get the rate from that. They match fairly fine with the data we have from rodents. That's the study I mentioned before in the wake rodents. So they measured both pre and post stimulus and also baseline. So we have the different rates for the different types as constraint, and we reproduce it in the circuit. Another nice thing that we reproduce, and is actually very relevant to our question of SST in depression, is what happens when you optogenetically silence SST. And the study in rodent showed that it leads to a twofold increase, a doubling of the baseline pyramidal rate, which really supports that hypothesis we talked about, right? So the less SST inhibition you have, the more the cortex is active at baseline. So we reproduce that as well. And more of a validation, as I mentioned, you see that there is some oscillatory signatures there in the raster. So when you look at the spike PSD, you see peaks at theta and alpha frequencies. This is 4 to 8 and 8 to 12, roughly, hertz. And that agrees with studies both in vitro and in vivo in humans. Taufik published a few interesting studies exploring the effects more in LFP, but really we were talking about the same bands, so theta and alpha bands, and that's a nice, it just emerged. We didn't really fit it. It's in every iteration of the circuit model, it was there. So that's fairly nice. And there are quite a few studies supporting the cortical generation of this rhythm. So you don't need external inputs to generate it. Obviously, external inputs might generate or modulate that rhythm, but there is definitely evidence that the circuits themselves intrinsically can produce this rhythm. So that's a nice support for the model. So now that we have this model that is reproducing quite a few things about human microcircuits, we adjusted it to apply some of the data we have about what happens in depression to generate depression versions of the model. So the first thing that I mentioned is this reduction in SST expression from our collaborators at TNCBIL. And we also used, because those effects are both synaptic and tonic. Tonic is more of the steady state inhibition, as I mentioned. Fortunately, we had some recordings of tonic inhibition in human pyramidal neurons from Taufik's lab, and we were able to use it to model those effects. So essentially, we have to generate a depression microcircuit model, we reduce the synaptic inhibition from SST onto all the other cell types, and also the tonic inhibition. And we calculated the proportion of that change in each cell type. So essentially, most of the effect would be on the pyramidal apical, but also SST inhibits PV to some extent and VAP. So some of this effect is there as well. And that's a very good point to mention, that even though you have a hypothesis of what the reduction in inhibition would cause, it's really hard to guess and to, I mean, like the circuit is so complex and cells are so interconnected that it's important to characterize it for real and not just, I mean, I don't think we can anticipate whether the reduction of a 40% would lead to a significant enough change in circuit firing properties and also processing. So that's really the setup of why we went about and did that. So now that we had the healthy and depression microcircuit models, we can start to compare and see what is the effects of this reduced inhibition. And so what we see is definitely an increased baseline in depression, and a significant one, about 50% Here you see the healthy in dark gray, and the purple is the depression. And we also quantified varying degrees of reduction going from 0 to 100. It's mostly linear, this change. As you can see, there are more spikes. It's very visible, that increase in the raster from 2 example microcircuits. So that was the first thing that we found, an increase in this baseline activity. But as I've said in the introduction, you need to see it in the context of processing of a signal response to gauge the full effect of that. So we also modeled the response in the circuits using that rodent cell type profile response paper, Gentet et al. We reproduced both the pre and post average firing and also the profile in each cell type. So pyramidals tend to peak right away and then settle down on a more lower persistent firing that is still above baseline. SST tend to be silenced, and that's done both by PV and VIP. And VIP and PV are activated early, as we know from some data in some of those papers. Their latency of activation post-stimulus is earlier than the other cell types. So we include all that. And this is an example raster plot of the firing in the circuit, which pretty much shows you the same thing, but more per cell here. I mean, you see cell 0 to 1,000. You can see the pyramidals firing and different populations. So now that we had a model of that, and by the way, just to, obviously the brain does many things. Those circuits do many things. There are different types of stimulus. It depends on the area, on the layer. The idea here is to look at a very simple example of response. It's a brief response. It doesn't, looking at a short window, about 50 milliseconds is usually what we analyze here. You see that the activity persists for about 100, but then settles down. The idea is also that later, 50 onwards milliseconds, that's the time that you might already get some feedback input from other areas, and that makes the whole picture much more complex. So that's why we are focusing on this brief stimulus and analyzing the 50 millisecond post stimulus, which is what you see here. So when we compared the response and the pre and post firing, as we saw before, pre-stimulus, there is about a 50% increase in the rate, but there is no change in the rate in the circuit post-stimulus, and that agrees with the hypothesis. So through the effect propagating through the entire circuit, it's mainly affecting the baseline and less the response of that kind of response. So health and depression, again, dark gray and purple, that's the pyramidal spike rate, which is our proxy of the readout from the circuit. So essentially the excitatory population, which is 80% of the circuit, also provides the sort of the more relevant readout for the next step of the cortical processing hierarchy. So given that there is not much of a change in response, but quite a change in the baseline, one measure that I mentioned before is the signal to noise ratio. So just really taking the ratios of those in each run, and you're looking at results from, I want to say, it could be 200 microcircuits, but at least 20 here, but I think it's actually 200. Most of the results you will see are from 200 random microcircuits, especially these. This is like from 200. I'll also mention, those are heavy simulations, but we've been fortunate to be able to run them on Synapse. It's a very nice resource of parallel computing that speeds up the simulations. So we see that if you take the ratio of those two, then there is definitely a clear and significant reduction in signal to noise ratio. So that again is a nice step. So we saw that there is an increase in baseline, there is a decrease in signal to noise ratio because the response doesn't change much. So that's definitely nice, and was sort of like supporting what we hypothesized would happen. And again, just to mention, the reduction need not, it didn't have to be significant. And I'll show you later how that is visible in some of the results. But the next step that was important for us is to further interpret and characterize what does that mean in terms of processing, right? Signal to noise ratio is one kind of measure, but we were interested to better interpret that. So what we did is the following. We looked at all the rates of those pyramidals pre-stimulus and also post. So pre-stimulus, we just scanned shifting windows and just checked the rate over 50 milliseconds. And that's the distribution you see here. Sorry again. And so you see the average is about, let's say, 0.6 hertz. And then there is sometimes when it's higher, like can even be 2 hertz in some sections of that baseline activity. And sometimes it's very low. And similarly, when you look at the rates post-stimulus, they can vary. And the reason they vary is for two main reasons. One is the background gives it a different context each time, so the background input, and also then it gets propagated with the stimulus differently through the circuit. Those circuits are not doing the same thing over and over, they're quite dynamic. So when you look at those two distributions of rates, response and baseline, a simple way to look at it is essentially to say, can we detect whether there was a stimulus or not? And, theory, established theory usually suggests putting the decision boundary at the intersection between those two curves. And that enables us to look at two important measures. One is the failed detection and one is the false detection. And this is really similar to failed and false positives, essentially. Or I would say it's a that will be here a false negative, right? So you sort of would say, no, there wasn't anything, but there was actually a signal. And here it will be a false positive, because you think, okay, there was a signal here, but there isn't any. So when you quantify that, and here in light gray is the failed detection, so you fail to actually detect stimuli because your decision whether or not to say that there was is right here. That's your threshold, and that's below the threshold. So you're missing those weak stimuli. And here, it's the false detection. So you think there was a stimulus, but there wasn't actually, right? And then, so this is shown for health. And then we can do it for depression as well in purple. And there is a clear change and increase in those failed and false detection rates. So this is summarized here. You see more than a doubling of those rates, essentially. So those microcircuits that have reduced SST inhibition and therefore a wider distribution of the baseline firing that blurs the difference between signal and response and increase those false and failed detection rates. And I mentioned about, the effect didn't need to be significant at any stage of the road, and it sort of, it shows clearly here. So if you look at different levels of reduction of SST, and then the probability of false detection on the left here and failed detection on the right, then going from the healthy level with no reduction of SST inhibition, you see, if you reduce just by 20%, you don't see much change in either failed or false detection. At 40%, you start seeing an effect, and that increases further, mostly linearly from that point on. But as you see, this is like a threshold effect, which at this time, I don't think we're pursuing it too much in terms of making a story out of it, right? Why? It's curious to think why at the level that is estimated by gene expression in depression patients, you see suddenly that jump in effect, But the main thing to illustrate here is, first of all, that there is a threshold, whatever it means. And the second thing is that a reduction of 40% need not have been significant, and that justifies looking into it and characterizing it to establish the link between the gene expression indicators and cellular indicators and cortical processing. So this is really pretty much was the end of that story. And as I mentioned, this is out on bioRxiv, so feel free to look at it. So to summarize, the main thing we find is a noisier cortical processing in depression. It's overactive at baseline. So that makes the distinction between signal and no signal more difficult to detect. There is a reduction in SNR, which is another metric, and consequently also a reduction in failed and false detection rates. And I'll mention that, interestingly, behavioral experiments in depression patients show similar results. So about a 2 to 3-fold increase in failed detection, and a larger increase in, sorry, just to get it right, it's a larger increase in failed detection compared to false detection, I think. So it's interesting that our models, even though obviously we need to be cautionary about their completeness in terms of capturing some of those things that underlie detection, right? I mean, we do not presume that at all, but they do actually agree with what you can see behaviorally. So that was very interesting to see as well. Another thing that we saw, I showed you today, is essentially those data-driven models of integrating human cellular circuit and also gene expression data. And lastly, that enabled us also to provide some depression microcircuit models, and those models can, I would say, also serve in studying other disorders. SST has actually been shown to be reduced in a variety of both neurological and mental health disorders. For example, Alzheimer, autism, even in aging to some extent. Obviously, it will be interesting to see what is the reduction level at each case, and how can that maybe explain some of the differences between the effects in each of those disorders. But obviously there is also a lot of similarity, and that could be explained by a shared mechanism. But yeah, I would just say that those computational models will be shared at some point openly once we publish it, and definitely encourage people to maybe make use of them for other kinds of questions. I'll finish up with a couple of future directions that we have, just to also mention a bit of how we further use these models in depression, especially study. The first thing is that because we have a biophysical simulation that actually is implemented in the LFPI platform that puts it in a volume, a physical volume, with a different head media going from gray matter to scalp and skull, we can simulate the EEG that would result from the circuit after filtering it according to established models that are implemented in this platform, and therefore try to identify biomarkers of those SST circuit changes and effects that could maybe enable high resolution new type of biomarkers. And that hopefully, if it is found, and we have some quite a nice progress about it, that maybe in a future talk I can tell you more about, but that with the aim of really improving clinical diagnosis, making it more precise, more objective, better able to utilize the EEG. Obviously, EEG is being utilized more and more in recent years for depression diagnosis. But there is definitely much more that can be done. EEG is a noisy signal. In many of the studies, they use machine learning methods that are pretty blind to whatever happens, the underlying machinery. And therefore, there is a pros and cons and definitely some limitations to those approaches. So we hope to be able to enhance that diagnosis and monitoring usage of EEG in that way. So that can be done by detecting certain patterns, but also maybe changes in the typical profile of the frequency bands in the EEG. Another thing is we're definitely looking to study cortical processing in mental health, using more complex examples and cases. So, this work that I told you about, it was more about brief stimuli and early response, but we definitely want to expand on that, and we've been doing some work on that too. And lastly, we have an ongoing collaboration with Etienne Sibyl and Taufik Valiante to try to test new pharmacology in silico using our models. So I mentioned about those alpha PAM as a promising new treatment method for depression that Etienne and his group have tested in rodents. But now the goal is really to translate it to humans. So the first stage, and that has been undergoing for a while now, and we have some interesting results. Homera recorded the effect of the drugs in vitro in the human slices on those alpha 5 receptors that are targeted by SST. So we want to model that and then incorporate it into our circuit models of depression and health, and to see the effects on the drug and also other references drugs, older drugs that are less effective, to get a better systematic characterization of those effects and establish this for a translation to humans. And we have already pretty much set the framework for this by having the models, having the way to gauge processing effects. So yeah, we're definitely looking forward to do that. And lastly, I'll thank my team first. So Alex and Kent have done a lot of the modeling work that you've seen, and Kent has led the questions about the SST effect on processing in depression. And that's in the paper that Alex and Kent are both jointly first co-authors on. Frank implemented the circuit in LFPI, which is the biophysical platform I mentioned, and he's doing a lot of the EEG biomarker studies and making a very nice progress about it. Sana is a new addition to our lab, and she is looking to adapt those models that are a bit more somatosensory centric to prefrontal with relevance to schizophrenia and audible processing, which he would like to model and then establish, using similar approach, the link between PV inhibition changes and the audible task processing in schizophrenia. And I would like to thank the collaborators, so Taufik and Homera, for the human data and a lot of very useful discussions. Similarly, Etienne and Thomas for the SST and drug-related work, and also part of the discussion group. John is a co-supervisor of Frank and has contributed, and Srijoy as well, with the curation of the human data and some of the interpretation of the gene expression data. John and Srijoy are from KCNI at KMH. Etienne and Thomas are from KMH. And as you know, probably Taufik and Homera are from your good Krembil Center. So with that, thank you everyone, and happy to answer any more questions.
Thank you very much, Etay. That was wonderful. Okay, so we're going to open the floor for questions, and if you have to leave, that's fine. And as I mentioned, Etay is happy to stay around. So I would like to start with my first question. Take a preference. So that was lovely, very interesting, and I like to hear the whole story. So I have just a small technical question about network size. So earlier when you talked about using sort of when I asked you about sort of that activity and how you sort of set it, I forget exactly how many cells were in your model, but it's probably enough that represents a slice, I guess.
Yeah, so we had, yeah, I'll actually go back to that. It's actually best even in one of those first slides. Yeah, so microcircuits is a bit of, there is a different definition. You can look at units that do something, right? But at least it's very useful. Usually people look at the column because, I mean, from orientation columns, it started the whole idea with Hubel and Wiesel, and they got a Nobel Prize for that idea. Obviously, the cortex is continuous. So I am definitely among the people that don't really delimit it too clear cut, but it's definitely useful to look at a piece of the cortex. Lateral connectivity is fairly distance dependent, which also sort of lends support to looking at modules that are, let's say 0.5 millimeter by 0.5 millimeter by the whole layers, right? So that's what we're looking at.
Okay.
And we're taking layer 2, 3 from that,
Mhm.
right?
Yeah.
Yeah.
So my question is really more is sort of a technical question, is that because we use sort of looking at hippocampus, as you know, is that when you sort of carefully sort of matched or optimized fitting the synapses and stuff, that's for actual cells. It's not as if you're looking at a scaled model. So you're actually, so I just want to confirm that. So it's as if you're really looking at a slice, not sort of scaling it because of the decreased number of neurons. So in some sense, is it fair to say that that background fitting that was done at whatever stage it was, it means like everything else that's coming from the rest of the animal, like so it's a good enough representation. So in some sense, your size, that the number of cells you actually use, kind of delineates what's enough, I guess, to capture what's, these changes that you're looking at in depression. Would that be fair to say? Because they're actual individual neurons you're modeling, not like a population because of the smaller network size. Is that correct?
Yes, that's correct.
Okay. Okay. So I just want to confirm, because that's really good to know. Okay. Thank you very much.
I'll also just say that, I mean, definitely we might be looking to expand to maybe have the full 7,000, let's say, in a layer, and also multiple layers. That's definitely an active line that we're thinking of, but definitely layer 2, 3 on its own, first of all, is an important integration unit. So it's meaningful. Plus for the EEG, because it's superficial, it affects the signal a lot. So for EEG signals, it should be pretty important and sufficient almost to capture. So yes.
Okay. All right. Thank you. Any questions? You can raise your hand or you can just speak up. I'll keep my eye on here. Oh, Julio, you have a question? Go ahead. Or sorry, it's Maurizio.
Yeah, it's me. Hi, Francis. Hello, Etay. I have to ask you a question. So I was very intrigued by this idea of the SNR that is actually increasing in the depression condition. So I was wondering if you could say actually a little bit on how you could include, or if you are looking into that, more on the transition between the healthy to the depressed stage. I mean, you show us how to get out of the depression, right? But how do I get into the depression based on your modeling stuff?
Right.
I mean, how do I make the SSST to fire less? What's happening? Do we have any ideas?
Yeah, definitely. So first of all, SNR goes down, right? So it becomes worse in depression rather than increases. I think you said increases, so just to clarify that. Yes, and the drug that, because of that finding and the hypothesis, Etienne's group at KMH engineered more specific drugs, right, that boost the inhibition, right? So if in depression you have less inhibition, when you give this drug, you're going to boost it back to more normal levels. And behaviorally, right, you see that the mice are getting more healthy in their, depressed mice would not explore the maze as much. They're more withdrawn and just sort of quiet and in their own head and sort of more stressed. But then with a drug, you counter that. So that's exactly how you would do it. So that's about the countering. I think your question also relates to another interesting point that we're seeing as well in our research, is which stage of depression are we relating to, right? And I would say that's a complex question. I think that will be hard to really pinpoint. We do see, at least for, let's say, the EEG biomarkers that I mentioned in the end, that they agree with I haven't told you much about it, but what we see in terms of the changes agrees with early depression, right? The other effects that are more in the mice are a model of chronic long-term depression. So it's definitely an open question, very interesting one. We have not yet pinpointed the stages that we relate to so much, but we are definitely looking to do that.
So just to clarify, this idea that I get a lower SNR implies that somehow the activity, the more noisy activity, eventually trigger the status of the network in some sort of chaotic attractor that facilitate the looping. This is what the remugination and all the stuff. This is the idea behind this.
Yeah, that's a good idea. Yeah, I think so. It's really that, I don't know if we can see it here essentially, but that the rates would be higher just when there is no stimulus given, right? It will be interesting to think about it in terms of actually getting stuck in right, in an attractor. We have not looked at that. I guess the way to do it would be to try to perturb the network. I think, that kind of comparison should give you an indication of what you would see, right? Because this stimulus is really a perturbation of the network. But yeah, it will be interesting to actually very explicitly try to perturb and see, does it get stuck? Are there attractors? We haven't analyzed it, but that is definitely a relevant question.
Okay, thanks.
Thanks.
Okay, I think, Scott, do you have a question? Go ahead.
Yeah, so sorry, my question was a little bit related to what Mauricio was saying, which is sort of about how you judge the effectiveness of these things, behaviorally, since depression is as a mental health disorder, it's a little bit different from, if you're looking at, epilepsy or Parkinson's or something where there is a very clear sort of neural correlate to the dynamics you're studying. So I'm wondering, and again, you sort of talked about this a little bit, but it sounds like from rodent studies, they've shown that the changes in network dynamics that you're seeing in your model are seen in rodent models of depression. So it's a reasonable assumption that these changes in network dynamics are indicative of the depressed state. Is that the right interpretation?
Yes. So the model is the rodents are a model depression animals. So there is a stress protocol, chronic stress, and then they exhibit a lot of the symptoms that you see in human patients in depression. It's about lower attention, lower detection of stimuli, more emotional and cognitive effects, also less exploration. Really, they don't feel like doing it. Exactly like, so it's a fairly decent model. I would also mention again that behavioral studies in humans examine the exact thing that we have characterized here. And it was actually nice because we only saw it after looking, for some kind of similar studies to what we show. So like we were not guided by those studies from the start, but we're happy to see that there are some signal detection and attention studies in humans that show a significant effect, right? So that I would say is also one way, at least in humans, to quantify and probe the effect. Yeah.
Awesome. Thank you.
Yeah.
Any other questions? I have one, but I don't want to hog.
I have one short question. Sorry, I didn't see the beginning of your talk, but you mentioned attention, and I was just interested in the fact that the cortex is very good at, during selective attention, modulating background noise. So do you visualize, is the results a sort of a static noise change, or do you think there's also an issue with the rapid modulation of signal to noise, particularly in ascending sensory systems, something the brain does very quickly all the time?
Yeah, I think that's a great question. So interestingly, and that's another line of investigation we're looking to do, those SST connections onto pyramidals are modulated by cholinergic inputs, right? And there is also serotonergic that is relevant to depression. So, and interestingly also, the human study that we used to constrain those models specifically even characterize the effect of cholinergic. So we even have it as like a cellular data in human. So that's definitely something we're looking to do and look into. And I think there'll be that extra dimension of not just the static change, as you say, but also the how maybe the modulation is affected. So yeah, definitely good question and looking
Thank you very much.
to, yeah, thank you.
Okay. All right. Maliha, you had your hand up. Go ahead.
Yeah, hi. I had a question about the baseline microcircuit activity. So,
Yes.
how the injection of, this background input, like in terms of implementing it in your model, how did you go about knowing that this is the right baseline, like we've achieved the right baseline activity? Because
Yeah,
was that solely based on, I think on your next slide, you
yes.
showed the spike rate. So that is what you were trying to match it to.
Yes, exactly. So we have this as a target or an objective, as we are used to think of. And there are some parameters that can be tweaked to establish that. One of them is the background input. Obviously, we're guided by physiological levels. So one of the things we do, for example, I used to, when I had the modeled small circuits, I used to have thousands of synapses, background ones on each cell. But that's, first of all, not good computationally in terms of runtime. So we replaced it by simple background mechanisms onto each dendrite. That gives you just a few of them per cell. That gives it represented background input pretty efficiently. So we have that. Yeah, so that's the main thing. And then we also tuned a bit the connectivity between cell types to get that target value. And fortunately, you don't need an algorithms to do it, right? There are certain things like the human fitting that sometimes to get those channel densities, you need to really run an automated algorithm, because if you had to do it manually, it's not very friendly, right? Challenge. Some people try to fit networks using genetic algorithms or other algorithms. That's a pretty intense optimization. Fortunately, we didn't need to do it. By tuning a few key things, we get the desired firing rates.
Thank you so much. Really enjoyed
Thank
your
you.
talk.
Thank you.
Okay, so I just quickly follow up on that before asking my other question is, so the distribution that you put on the dendrites of the synapses was based on data or like experimental data, or yeah.
Yeah, so good question. So just to emphasize, there is the background and there is the intra-circuit kind of synapses, right? So the background is more simplified. The intra-circuit ones have some rules about, let's say, SST always activating the apical dendrites of pyramidals, right? PV always activating the basal, and then the rest of them, there is the logical rules, like VIP is not gonna really connect to peer because there is no evidence really that they do that, right? And yeah, so that's sort of it. In terms of more fine scale rules, some people sort of have distributions. We didn't do it. It was pretty random.
Okay.
On top of those rules, it was random.
Okay. All right. Okay. So the question I had was more about was about the SST. And as you know, and you have experts in your lab about, cell type specific. So SST, of course, is not one cell type. And so just in terms of the data that you guys had, be critical, because that's kind of what you're basing, in the depression data. So what is known about this SST cell type? So is it that you're assuming they're Martinotti, and what is known about other SST positive cell types in that layer, or is it they're known? Yeah.
Yeah, so that's a good question. markers like SST, PV are not one-to-one, they're not unique. Sometimes people even use a different marker. I think it's the cal binding, or either, I forget if it's the calretenin or cal binding. There are other markers that sort of give you, are a bit more uniquely expressed in Martinotti. So I think definitely we are sort of looking more of the what cell type wise was defined as Martinotti, right? So Martinotti, for those of you who don't know, those would be interneurons that have axons that go all the way to the superficial parts of the cortex and pretty much target the distal apical of pyramidal. So you can really distinguish them by the shape of the axon and also by the adaptive firing. So when you look at interneurons, it's not that hard to actually say, okay, this is a Martinotti. Fortunately, there are those combination of indicators that give you this. The reason why we talk, especially more in recent years, about SST is that in those cells, the Martinotti, there is also, they tend to be distinguished by a larger and more unique expression of SST. I would say for this forum, there is a pretty good equivalence. Obviously, there are some other, even PV, I think, might express a bit of SST. I'm not like, I may not be that accurate, but there is definitely a bit of ambiguity there. But yes, it's
like, so for example, like I was kind of wondering about the correspondence, how much is known. So like the synaptic data, the disynaptic human one you showed, which was SST. So it was, what was known was it was just SST as opposed to, this one, for example.
Good question. Yeah, good question. So this cell was established as a Martinotti using firing, because they also give a pulse and see the firing and the axonal. I don't know if they even checked it in terms of RNA or gene expression for SST. So that's actually worth checking, but that, so
and then
it is,
and then the Etienne's data is SST, but that's at the genetic level, so that obviously can't really say much there.
Right. But the nice thing, it's SST in SST interneurons.
Okay.
So it shows you that their machinery, the cellular machinery, is compromised, right? So that's, and just as a note, sometimes you see a bulk expression. You take a whole piece of cortex and you see a reduction in SST. That doesn't give you usually a layer resolution, and it doesn't give you a cell type resolution. But nice thing about this study is fairly focused.
Okay.
How to translate this into modeling, it's challenging, right? I mean, there could be alternative ways to do it. For example, there could be maybe just synaptic loss rather than weakening of the synapses. But overall, our assumption is that the net effect on inhibition would be the same, but we might actually be looking into examining some alternative modeling translations of this gene expression data. But just as a word, that's not a trivial thing. Yes.
Yeah. So when you guys did that model, you just sort of you removed the SST from the circuit, right? I guess, is that
reduced?
So when you were doing the depression, like simulating the depression with the SST, you sort of just removed the SST cells from the circuit, meaning
no, because interestingly, actually, in the TN study, they also quantified the number of SST interneurons. This does not change in depression. So, right, so there is the same number of them, but presumably their synapses are weaker.
Okay. So that's what you changed in the model. Yeah.
Conductance, synaptic conductance from SST to other cells, and also overall contribution to tonic inhibition.
Okay. Thank you. And I see Alex has said these are putative. Yeah. Okay. I'm not sure.
In some of the other slides, these are more putative, especially, like this. This is the putative SST. We did correspond with the Allen brain people, and they spend a lot of time actually categorizing and thinking about those things, right? And they confirmed that this should be a Martinotti, that specific ID. And so we refer to it as SST, but as Alex said, it's a putative SST, yes.
Okay, great. Okay. Any other questions from people here? You can just speak up if I don't see hands. And I see Michael just entered the room. Okay. Okay.
So maybe I can ask another question. Sorry.
Go ahead.
Sorry, I let you go eventually, but I was very curious. So you mentioned at the beginning the important, so these remarkable phenomena about the synaptic facilitation. So one single input essentially from the pyramidal cell laterally can trigger the SST. So a twofold question. What do we know about the short-term plasticity on the inhibitory inputs to the pyramidal cells, if there is any, if there has been any kind of classification or characterization, and how this short-term plasticity actually plays inside your model. So
let me just get there. So the short-term plasticity here, right? That would be there is a nice model about it that has been around for quite a while that has a few parameters. The success rate.
Misha?
Misha, exactly. Yeah, Misha Tzodik's, it's the UDF. So there are three parameters: the use, the facilitation, and depression time constants. So we have that, these parameters, and we tune them to reproduce the observed short-term dynamics as much as possible. For this, we had data. So we actually capture the human short-term dynamics of this connection, right? For the other ones, there isn't enough data, but I've been actually part of a recent human modeling workshop that they talked about upcoming data about those things. So we will have that for other synapses as well. And your question, was there any other part of the question that I have not answered?
Yeah, so what happens if you take out this facilitation, or you just take standard synapses? How is it changing the result?
That's a good question. So like, if there is anything that will cause that, what will happen is that you wouldn't spike really, right? The summation would be sub that. Don't want to say linear or not linear, but it will be below that. And then essentially you wouldn't spike, you would require more pyramidal cells, right? And you would really weaken the lateral inhibition, which at the end, Sibyl likes to look at it as a blanket of inhibition in the baseline that keeps things sort of like steady. So you would lose, you will create, like it will be less powerful, essentially.
I see.
And that should affect as well the signal to noise processing detection, right? That will, yeah, that's actually an interesting question. I don't know if there are specific mechanisms or things that affect those temporal dynamics. I haven't, I don't have the experience in, looking at it in terms of actual mechanisms. But if there is any change in any disease in those mechanisms, it would have quite significant effects, like reduction of SST inhibition, essentially.
Okay, thanks.
Thank you.
I also have a question. Let me open my.
Go ahead, Alarisa.
Yeah. For this figure exactly, you have intracellular recordings of human, if I'm right.
Yeah, it's a paper. This paper characterized this thing in humans, and we reproduce it.
And is it in vivo or?
It's in vitro.
Vitro.
Yeah.
Got it.
You cannot do this thing in vivo right now.
Yeah, I know. Yeah, that's why I was wondering.
Yeah. The nice thing about in vitro, at least, and that's Taufik is doing a lot of in vitro work, is that it helps you tease apart some of those properties which remain as they would be mostly in the intact brain, right? So, yeah.
Thank you.
Thank you.
Okay, and looks like we're just about 11:30. So does anybody else have any burning questions? Okay, if not, let's thank Etay or Dr. Hay again for an excellent talk, and maybe we'll have you come again when you have more results.
It will be a pleasure.
Thank you very much. So if you gotta go, you gotta go. And maybe just anybody wants to say if they have anything else burning, Etay could just stay for a short time. But otherwise, thank you everyone for attending our KCN event. Okay, bye. Yeah, that's really cool, Etay.
Okay.
Yeah. I mean, I actually one thing I'm just wondering is about, like, as Maurizio was just asking about the short-term plasticity, right? Because you're probably aware, I think it's Kempter, other people, where they sort of look at these relationships. I think Alex probably knows about this work, with the the short-term plasticity in specific ways affecting, the output of the circuit. So I don't know if you guys would you try to focus in that way in sort of reduced or simpler models to sort of get a handle on that? In
Yeah,
terms
I
of how
must
it's
say,
contributing. Yeah.
yeah, I must say that those questions pretty much opened my eyes a bit to that direction, and that's nice. Homera is seeing some drug effects on time, I think time constants. It still remains to be, analyzed further, but we are looking at some of those effects as well, which could essentially affect the summation properties, right?
Mm-hmm.
So we could see, yeah, it will be hard a bit. Like the drug, I think, study would be the most relevant because, like, when you think about it, you don't have in vitro human data, electrophysiology from health and depression so much, right?
Right.
Nobody has focused on that,
Yeah.
but that's a possibility,
Mhm.
definitely.
Yeah. To make the link these levels. Okay. All right.
Yeah.
Yeah, that's cool. I see Milad there. Sorry, go
Yes,
ahead, Milad.
yeah, actually, I just sent you one email right now. I don't want to repeat myself, but this is a good thing because Alireza actually asked this question because Alireza's project is kind of related to inference of short-term, dynamics of short-term synaptic plasticity. And we just use in vivo data. And as you said, this is kind of something, it's, theoretically, it's not possible as in vitro because you have postsynaptic